Hey, what's up, classmates? So today we're going to be talking about amino acid catabolism. I'm sorry I'm recording this a little bit late. My family really do be loud, and I had to wait for a good time to record. So before we actually even start the objectives, what is amino acid catabolism? It's basically just a way that we use amino acids to create energy for us in the form of ATP or in a form that enters the citric acid cycle and then make redox currency and then make ATP or make sugar, make glucose from. And why is this important? In states of starvation or in times where we have excess amino acid and we don't know what to do with it, we can use it as energy. So there's kind of two themes with the two objectives today. How do we use amino acids for energy or gluconeogenesis? And the second theme is how do we do so safely? Because amino acids have an amino group, so a nitrogenous group, which is toxic to us. It's a threat basically to our brains, and we'll talk about that a little bit later. So yeah, two major themes: how can we use amino acids as energy, and how do we do it safely? So the first objective is be able to outline/flowchart the general pathway for the breakdown of all amino acids and where does each step occur in the body. And so there's three major sources where amino acids can come from. It can come from our diet, it can come from breakdown of our muscle, and it can come from other tissues apart from the liver where amino acid is abundant and is not being used, so where it's in excess. So essentially, the first way, dietary protein, that's going to come from our GI tract, and and the book goes into great detail how we do this, but the objectives don't say that we need to know. So in summary, pepsin in our stomach, pepsin combined with an acidic environment is going to denature the protein that we ingest, and then it's going to eventually travel to our small intestine or duodenum, and it's going to further cleave these proteins into individual amino acids. Once this happens, our small intestine is able to uptake these amino acids into the bloodstream, taking it to our liver. The other sources, they also travel to the liver, and this is because most of amino acid metabolism occurs in the liver, and that's what I have written here for you guys. Here's another picture that shows us more specifically how it's done. So amino acids, they're broken down when we eat them, and we're ingested, and then they travel to the liver through the bloodstream. Amino acids coming from the muscle, however, they come in the form of alanine or glutamine. And then the third way that we can use amino acids is from other tissues, and other tissues also use the form of glutamine to travel to the liver. So in summary, protein from the diet enters liver as amino acids, protein from muscle transports to liver in form of alanine or glutamine, and protein from other tissue is transported in the form of glutamine. All right, so second objective, be able to draw the structure of PLP and outline how it works in transaminases. Pretty weird how this objective was written, so let's talk about transaminases first and what they are, and then let's talk about the coenzyme afterwards. So basically, transaminases, they're going to catalyze this reaction right here. I can't fit it on the entire screen, but here. Okay, and so we're going to start with an amino acid, right? And this amino acid is going to have a amino group right here. There's this red NH3+, right? And then we're going to start with alpha-ketoglutarate, and if you don't remember where you've heard that from, it's from the citric acid cycle. And our alpha-ketoglutarate, alpha-keto, in our alpha position right here, we have a ketone group. And what's going to happen in this aminotransferase reaction or transaminase reaction, these are interchangeable, they mean the same thing, is an amination of our alpha ketoglutarate group, but then a deamination of our amino acid group. What does that mean? They're essentially just going to switch places, right? So we use our transaminase enzyme, and this amino group is going to find itself on alpha ketoglutarate right here, and it's going to turn into glutamate, all right? And then our amino acid is going to get a ketone group in place of that. So notice how the blue and the reds have switched, and now we get a alpha keto acid. Now this is important because in order to use amino acids as energy, we need a way to excrete this amino group safely, and we do that through the urea cycle. And so the urea cycle uses glutamate as a precursor, as one of the major precursors. And so that's essentially what's happening, is we're transferring this amino group from one amino acid and creating a more usable amino acid with this amino group. I hope that makes sense. And the coenzyme is PLP. And this stands for pyridoxal phosphate. It's just a coenzyme, basically. How it works, it's an intermediate carrier of the amino group in transamination process. And so in this process, it's basically this amino group is going to first be accepted by the PLP, which will then transfer to our alpha ketoglutarate to make glutamate, if that makes sense. So this is what it sort of looks like. One important thing, this pyridoxal phosphate, it's attached to our enzyme via a imine link, I-M-I-N-E. If you don't remember what that is, it's from OChem. It's just a N double bond C, and that is also known as a Schiff base. So that's all this is saying right here. So imine bond, Schiff base, and then this guy is the intermediate used, or coenzyme used to create an intermediate to allow for this reaction to happen. And the way it does it is a carb anion stabilization. So the intermediate will have a negative carbon charge at one point, but this coenzyme basically stabilizes that a little bit, makes the rate of reaction faster. All right, so objective 3, be able to describe with examples and structures why glutamate, glutamine, and alanine are so important in human nitrogen metabolism. So this is once again kind of going back to the threat of using protein as an energy source, and that's amino groups. And so we'll see here later that glutamate and glutamine are important because these are the forms that we want our amino acids to take in order to successfully metabolize our amino acids in a safe way. I know that sounds kind of confusing, but it will be cleared up later. So describe why these are important. So let's just do each of them. So this first one is alanine. You're going to use alanine. You're going to use a transaminase, right? And you're going to switch this amino group, get a keto group here, and this keto group here will get an amino group here. And this is glutamate, right? So we always make glutamate whenever we do this transamination, right? And what is this group right here? Why is this important? So if you don't remember what this is actually pyruvate, right? From glycolysis and and the Krebs cycle, right? And why is pyruvate important? Well, it can enter, right? It can become acetyl-CoA, and it can enter, right, the Krebs cycle, right? And once it does that, it can be used to make energy, make redox currency that can be used in the electron transport chain, right? Or, right, if it enters this citric acid cycle, remember in the citric acid cycle, one of the intermediates was oxaloacetate, and this can enter gluconeogenesis, right? And so pyruvate can be used eventually in the long run as gluconeogenesis to make sugar, or it can enter the citric acid cycle to make redox currency for electron transport chain and ATP production, right? And what was this guy again? Do you guys remember? Oh, this guy is alpha ketoglutarate, right? Alpha, so C1, alpha beta position, alpha ketoglutarate, CH2CH2COO, all right? And then what was glutamate used for again? We sort of talked about it earlier, but this is an important precursor amino acid in amino acid metabolism, because we need this guy essentially to successfully make the products that are needed for safe amino or nitrogenous substance excretion. I'm going to keep repeating that. It might not make sense now, but eventually, hopefully, it will start to make sense. So alanine is important because it can produce pyruvate, which can enter the citric acid cycle, and it can be used as gluconeogenesis precursor. All right, glutamate is okay. So this is glutamate, and here we just show that it can become alpha ketoglutarate, and alpha ketoglutarate is another precursor in the Krebs cycle. So this can also go into the Krebs cycle, right? And this can also enter ETC, which eventually makes ATP, right? Or this can also go into gluconeogenesis. All right, and so, yeah, we're going to use the glutamine synthetase enzyme for this instead. We actually use redox currency here, but a different type of redox currency. And actually, we can actually use NADP or NAD8 or NAD+. So either works, right? And notice here, this is also redox currency. This process here is also redox currency, right? Which can enter ETC, all right? And what did we say earlier? This is an important precursor in the urea cycle, all right? So not only does this process right here make a reaction that can enter the Krebs cycle, but also this is an important precursor for successful excretion of this amino group in amino acid metabolism. So similar to alanine, nitrogen metabolism of glutamate can produce Krebs cycle intermediates, which can yield energy or enter gluconeogenesis. Also, right, ammonium ion is excreted in the liver and can enter the urea cycle. So we've mentioned that multiple times already, right? Glutamine, very similar to glutamate, but it holds an extra amino group. It holds an extra amino group, right? And so you can attach another amino group right here, right? And you can transfer it to the liver that way. And now you have two, right? This and this. You have two amino groups that can be excreted in the liver. So it's kind of more of a compact way to send these toxic amino groups to the liver, but in a safe way, if that makes sense. Hopefully that makes sense. All right, so we're gonna jump to objective 6 here, guys, because I like this order that I'm doing it in. It makes more sense to me chronologically than the order that was presented to us. So I think we did like 1, 2, 3, and now we're doing 6. What is glucose-alanine cycle, and what is its significance in an active body? So similar to how glutamate and glutamine plays a role in transporting amino groups in a non-toxic form to the liver, alanine can do the same thing, but alanine can enter a cycle where it continually shuttles your amino group, and it does this by using pyruvate and glutamate and alpha ketoglutarate to do this. So here is the cycle, right? So we have glutamate, which is turned into alpha ketoglutarate, and pyruvate, which is turned into alanine. So if you remember, we actually went this opposite direction in the previous slides or a previous objective. Let's just show it here real quick. So this right here, right? Sorry, not that one. Oh yeah, we can use this one. So pretend this amino acid is alanine. So we went this way, right? We went forward from left to right. Actually, this is a reversible process, right? So let's draw arrows like this. And so it can either go this way or this way. And in the alanine pyruvate cycle, it's actually going in the reverse direction. And this is possible because this is the reversible reaction, right? So let's go back to our objective here. And so we're using alanine aminotransferase, and instead of alpha-ketoglutarate to glutamate, we're getting glutamate to alpha-ketoglutarate, and instead of alanine to pyruvate, we're getting pyruvate to alanine. And so then alanine can be sent to the liver, and then this same process, the forward process can occur, and then we can create glutamate, right? And then glutamate here can, remember, it's an important precursor. It can excrete that amino group safely in the liver, and that can be turned into urea, and urea can be excreted through our liver, all right? And so this is basically just a cyclical shuttle system with alanine and pyruvate, taking advantage of the fact that this reaction is reversible, right? And it's a way to basically just excrete this urea, all right? So I'm just going to reiterate, in order to do anything useful with amino acids, we have to get rid of the amino group, or ammonium ion. This is a hassle as it can only be done in the urea cycle, which occurs in the liver, right? Okay, so now we're going to jump back into the objectives. What are the 5 entry points into citric acid cycle for carbon skeletons from amino acid degradation? And so this is the whole point of amino acid catabolism, right? We're making things that can enter the citric acid cycle, which then produce redox currency that can power our electron transport chain, which produces energy for us, right? And so there happens to be 5 places that we can do this. Yeah, and we've already talked about 2, right? So we've already talked about 1 that turns into pyruvate. We literally just talked about it. If you don't remember, that's okay. It's alanine, all right? Alanine. And the other was alpha-ketoglutarate, right? And what was that? That was glutamate, right? Glutamate can be turned into alpha-ketoglutarate, right? And then there's 3 others, right? So like, for example, valine can enter can become succinyl-CoA, right? Or for example, tyrosine, right? Tyrosine here can be turned into fumarate, right? Or valine can be turned into oxaloacetate, right? So these are your 5 entry points: pyruvate, oxaloacetate, fumarate, succinyl-CoA, and alpha-ketoglutarate. But there are a lot of examples of this, okay? Tons of examples. So not only alanine can turn to pyruvate, not only valine can go into oxaloacetate, but many others. And so here's just an example of one, right? So aspartate and alpha-ketoglutarate. This is going to turn into glutamate, right? And aspartate is going to turn into oxaloacetate, right? Oxaloacetate, right? And this is just using that transaminase reaction, right? And then this can enter the citric acid cycle. One example, right? But there are many amino acids that can be used in multiple areas. So everything you see in orange here, those are all the enzymes that can enter the 5 sites that we talked about. All right, on to the 5th objective. What is meant by glucogenic or ketogenic amino acids? Be sure to give examples using structures. Essentially, your glucogenic amino acids are the ones that can be metabolized to enter your citric acid cycle for either energy production or gluconeogenesis. Here's one example. Serine, when we use serine dehydratase, right, turns into pyruvate, which can enter gluconeogenesis to make glucose, right, or can enter the citric acid cycle to produce ATP. Ketogenic ketogenic acids are just the amino acids that are metabolized and can be used to produce ketone bodies. And so these guys, they were used, and these guys can enter the citric acid cycle, which can then enter gluconeogenesis, right? Well, these guys can be used to make ketone bodies. And one example is phenylalanine, which turns into acetyl-CoA, which is a ketone, right? And this is a multi-step process. We're not going to go over it. The objective says specifically that we don't need to know every single reaction because there's a lot in the textbook. If you want to learn them, go ahead. I am not going to do that. So yeah, there are 7 amino acids that are ketogenic and 19 that are glucogenic. And then 5 of these can actually be both. So 5 of the amino acids can actually be used to create ketone bodies or enter the citric acid cycle. And so here, earlier we talked about the pink boxes, right? These are your glucogenic. Well, these in the blue are your ketogenic. All right, guys, last objective, objective 7. So we did 6 earlier on, remember? So now this is our last one, number 7. Why is a balanced diet essential for protein catabolism? What are the coenzymes important in protein catabolism? And be able to describe the 1-carbon transfer reactions, including the roles of important cofactors and coenzymes. So why is protein catabolism important in the first place? It's used for energy, right? And actually accounts for 10 to 15% of energy, which isn't a lot, but it is substantial, right? And so it is important that we have energy, or we do consume protein to use for energy in our diet. But too much is also detrimental. And why is this? Because remember, whenever we metabolize protein, we increase the concentration of ammonium ion that's present in us. And this guy is actually very potent, very toxic, right? And we'll talk about this in the next video, the urea cycle, why it's toxic. But yeah, essentially it can damage your brain. And we'll talk about how so in the next video. Yeah, so what are the important reactions in protein catabolism? The one we really need to know is this protein deamination. And that's what we've been talking about this entire video. And that's going to use, right, our paradoxal phosphate as a coenzyme. And you're just going to switch your ketone group from one substrate with an amino group, and then an amino group from another substrate with a ketone group. You're just going to swap those positions. We went over that earlier in the video. So apart from that, there's one other reaction that the book mentions, and we don't have to know this to the fullest extent, but we have to be able to recognize it when we see it. And this is a 1-carbon transfer. Some examples are page 692 and page 693 in your book. So if you can just recognize what's happening, then that's great. So there are 3 important cofactors for these types of reactions. They are listed right here: biotin tetrahydrofolate and S-adenosylmethionine, or SAM for short. And these are the structures. We need to know how to recognize them. We don't need to know how to draw them. But yeah, so that's all for this first objective set. The next one will be urea cycle. That one is going to talk about that second thing we talked about. Remember in the very beginning, whenever we use energy, we have to do it in a way that's safe, because this ammonium ion right here is very toxic to us.